We'll start with today's PMRF-NPTEL session on bioengineering and interface with biology and medicine. This is the week 4 PMRF-NPTEL session. PMRF-NPTEL sessions are designed in order to bridge the gap between the course members and the students, and these are interactive problem solving sessions in which the PMRF fellow associated with the course, such as myself, takes interactive lessons with the students who are opting for the course. And in these particular sessions, we sit down and solve the previous year's assignment problems of that particular week. This will help students get a better insight into how exactly to tackle the assignment questions at the end of each course, and have a better understanding of the entire course itself. So let me introduce myself. My name is Anubhav Chatterjee. I am a PMRF fellow and PhD scholar at the Developmental Neurobiology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. So for this, the week 4, we have gone through the basics of genetics and Mendel's laws, and we are going to have a session on that and are going to just try and solve various problems related to genetics. So let us get started. So this is a condition type of question. Our condition is a JRF Imtiaz started working on a rare species of pisum, which is essentially pea plant, for understanding the genetic hallmark theories. He took 1,000 pea plants of various traits, green wrinkled, the capital G and smaller. So from this itself you can understand that green is the dominant trait, because dominant trait is always given in the capital letter, and wrinkled is a recessive trait because it is in the small letter. 250 of those, green and round. So round is our dominant trait in the second one, which is given in the capital R. 250 of those, yellow wrinkle, 250 of those, and yellow and round, 250 of those. So, but one thing to see is that all of them are homozygous in the traits, which makes life a lot simple while working on the problems. So we have to answer the following questions based on the condition given above. Imtiaz took green and yellow pot colored plants to observe and understand the difference between genotype, phenotype, and alleles. Which of the following statements are true? Pick the correct combination. The options are: green and yellow pot colors are GG and small gg phenotypes. No, okay, so green and yellow are pot colors, and GG and small gg are phenotypes, and both strains are homozygous. Capital GG and small gg are alleles, and green and yellow are pot colors are genotypes, and none of the strain is homozygous. Green and yellow pot colors are genotypes, and capital GG and small gg are phenotypes. Only yellow strain is homozygous, and capital GG and small gg are genotypes. Green and yellow pot colors are phenotypes, and both strains are homozygous. So since this is only one type of correct, one option correct type of answer only, you can put your options that you think are correct into the chat. Archana says it is D, that is capital GG and small gg are genotypes. Green and yellow pot colors are phenotypes, and both strains are homozygous. Someone else wants to try? You can put in what you think is the correct answer into the chat box. Shreya also says it is D. Okay, so let us look into the solution slide for this. So what is exactly the difference between genotype and phenotype? This is a very basic question that will get started into the entire discussion on genetics. But first, before genotype or phenotype, let us look into what is a gene. The Mendelian gene is a basic unit of heredity, and the molecular gene is a sequence of nucleotide in DNA that is transcribed to produce a functional RNA. So when Mendel did his work, he described these as factors that are inherited between various progenies. But nowadays we know that the term actually is gene, and for in the case of Mendelian genetics, we still believe gene, we still call gene to be the basic unit of heredity that is passed from generation to generation. But in a molecular sense, a gene is a sequence of nucleotides present on the chromosome that actually codes for a functional RNA. That is, it can be transcribed. And since it can be transcribed, there can be two types of genes. One is a protein coding gene and a non-coding gene. Protein coding genes get translated to form a functional protein, whereas non-coding genes only form a RNA and do not get translated to a protein. So, can anyone give me an example of a non-coding gene? So, protein coding genes are ones that form functional proteins at the end of the entire transcription and translation process. But okay, Archana says it is telomere. That is okay. So telomeres are stretches of DNA that are present at the end of the chromosomes. But Archana, the thing is that are not transcribed. Telomeres do not get transcribed into RNA, so they essentially do not even fall into the category of genes. They are just interspacer DNAs that are present there. Since they are not transcribed, they do not fall into either of the categories of protein coding or non-coding genes. So what you have to think is that which very famous RNA, so which very famous RNA does like does its function only in the form of RNA? Does not like it does not need to be translated to form a protein in order to function. I think most of us have an idea of which RNA I am talking about. An RNA which a functional RNA which does not need to be translated into a protein. Ribosome, yeah. So I am talking. So ribosome is a like closely looking at it. We are talking about the ribosomal RNA. Ribosomal RNA fall into the category of non-coding genes because they do not form a protein. So that was the difference between the two. Now we move on and look into what exactly is an allele. Allele is something that is thrown around a lot during genetics. So at a chromosomal or genomic location of a gene or any other genetic element, the chromosomal or genomic location of a gene or any other genetic element is called a locus, and alternate DNA sequences at a locus are called alleles. Essentially, alleles are different forms of a particular gene. So a particular gene can form two different RNAs, and these RNAs can be translated to form two different proteins. And these alternate forms of the gene of the same gene is known as an allele. So that is exactly what the difference between gene and an allele is. Allele are alternate forms of genes. But what is exactly the difference between a genotype and a phenotype? Genotype is the organism's genetic information. This is something that we never get to know when we look at the organism. Whereas a phenotype is the observable traits. So genotype essentially are the alleles or the nucleotide sequences. Phenotype, on the other hand, are the various physical traits such as the height, the color, if the seed coat is wrinkled or round, if the flower is axial or terminal, things like that. That is the phenotypic traits. So now there is since there's this huge difference between genotype and phenotype, there is also one interesting thing that happens. There can be two different types of genotypes. So if two different alleles are present together, one allele will dominate over the other one and be represented into a phenotype. So if two different alleles, say B and B, are the two different alleles, capital B and small b, if they are present together, only one of them will be expressed in the phenotype. The one being expressed is known as the dominant allele, and the one which is suppressed is known as the one that is being suppressed is known as the recessive allele. And the dominant allele is given by capital B, the recessive one is given by small b. So in case two capital B alleles are present in the genotype, then we get a dominant trait such as purple. If two recessive traits are present such as small b's, we get a recessive phenotype, that is white. But if both of them are present together, that is capital B and a small b, we still get a purple colored flower because the capital B phenotype dominates over the recessive smaller b. But so in case of the phenotype, we'll see purple in two cases and white in only one. But when we see it in the case of genotype, there are exactly three different genotypes in all of the three different cases. And when the alleles are the same in the genotype, it is known as homozygous. So capital B is a homozygous dominant. Similarly, capital small b is a homozygous recessive, and both of them, if present together, is known as heterozygous. So this was essentially the difference between a gene and allele, genotype and phenotype. Does anyone have any questions regarding this? Okay, I guess not. So we can move forward with our discussion. So depending on our discussion here, for the particular question, if Imtiaz to green and yellow pot colors and want to differentiate between genotype, phenotype, and alleles, the correct option is D, as everyone also pointed out. That capital G and small g denote the genotypes. Green and yellow pot colors, which are observable traits, are the phenotypes. And both the strains are homozygous because when present, capital G and capital G are homozygous dominant, and small g are homozygous recessive. Both are homozygous because both are the same type of allele, dominant or recessive. So that was that question, and now we move forward. So now we are getting into our first question on genetics, the problems on genetics. So most of them are we have to draw our Punnett square and find out the various solutions. So I would very much encourage everyone to get a pen and paper, or whatever you want to scribble on, and try and solve these problems alongside me so that you get in hang. So get anything you want to write on, because we have to draw a lot of Punnett squares throughout the entire remaining of the questions. So the first question is, in the first experiment, Imtiaz took a yellow round and yellow wrinkled plant. He performed a cross between yellow round and yellow wrinkled plant. Which of the following is true for the F1 generation? F1 generation is the first filial generation or the first progenies of the parents. The options are: the genotype of the F1 generation will be G, small g, capital R, small r, small g, small r, in equal ratio. The genotype of the F1 generation will be this for all the progenies. The phenotype of F1 generation will be yellow round for all progenies. The phenotype of F1 generation will be yellow wrinkled and yellow round for equal ratio of the progenies. This is a box type of answer, so there are more than one option that is correct. So let us I will help you with the first part. So yellow round, the first type of plant he took, the genotype is small g, capital R, and yellow wrinkled means the genotype is small g, and small r. This is the parental generation of the P generation. We have to go ahead and find out what the situation is in the F1 generation. So this is a dihybrid cross because there are two traits we are talking about, yellow and round, like the color and the seed quality. So everyone should get their, like, try and go through this. Like, what do you think the genotype in the F1 generation should be? Archana says all are yellow round. Okay, so we have one answer. Anyone else that wants to try an answer? I just go ahead and draw a Punnett square. So for these type of dihybrid process, I encourage like my way of doing it is not exactly drawing the entire dihybrid cross with all four traits, but since we know Mendel's law of independent segregation, we know that these traits small g and these small g, that is the color traits, will segregate separately and the quality traits will segregate separately. So if we draw a very simple 4 by 4 2 by 2 Punnett square for the seed color and a separate 2 by 2 Punnett square for the round, then oh wow, we have a lot of ones. So separate 2 by 2 for the seed quality, then we should have like simplify the entire problem instead of drawing a big 4 by 4 Punnett. So we have more answers. So Archana had already suggested that all are yellow round. Arushi also says that the genotype is small g, capital R smaller, and all are yellow round. Nidhi also says the same, that small g, and capital R smaller should, will be the genotype for all F1 progeny. So let us solve this. So here we have the Punnett square for the colors. So since one is small g, and the second one is also small g, so all of them will be small g and small g. So homozygous recessive yellow. Secondly, for the quality of the seed, we have R and R on top, small and smaller on the left hand side. Punnett square reveals that the all the genotypes should be capital R small. So we have small g and capital R small r from the cross in the F1 generation. So everyone was correct in calculating this. So our correct answer is the genotype of the F1 generation will be small g and capital R smaller for all the progenies. And since this is a multiple choice question, this should also have this as an answer. And not this yellow round. Oh. So everyone was correct. The progenies for the F1 generation all are small g, capital R, small r, and the phenotype, as everyone has suggested, should be yellow and brown. Because yellow, because it is homozygous recessive still, so that will be yellow, and round because the dominant big R trait allele dominates over the smaller one, a recessive one, and gives us a round progeny. So that was the correct answer. Anyone wants to ask anything from this? Okay. So we go back into the go forward into the next question, which is Imtiaz has given was given a single blinded plant with phenotype yellow round pot features. Single blinded means that you do not know the genotype. He needs to tell the correct genotype of the given plant to the farmer who brought the plant for testing. Which of the following methods should he use to decipher the genotype of the plant? So Imtiaz was given a plant with whose phenotype he only knows, genotype he does not, and he wants to know what's the genotype of the plant. So which of the following techniques can he use? The options are self cross, test cross, back cross, or the genotype is small g, capital R, which is confirmed. Arushi says it is test cross, that is option B. Everyone, I would encourage all of you to try and guess which should be the correct answer. Shreya also says it is a test cross. Anyone else? Ria also says it is B, it is a test cross. Okay, so a lot of answers. So everyone is suggesting that it should be a test cross. So let us go ahead and look into what exactly a test cross is. So everyone was correct, it should be a test cross, but let us look into detail what exactly is a test cross. Test cross is a cross between organism with dominant phenotype and with a recessive. So when you look at a particular trait and you observe a dominant phenotype, the genotype can either be homozygous dominant or can be heterozygous dominant. So capital D or capital D small d. So in order to find out, but if it was recessive, you can just look at it and tell that it was a recessive phenotype means a recessive genotype because no other combination is possible. If the phenotype is recessive, you can just go ahead and say that the genotype is homozygous recessive. But in case of phenotypic dominance, you have to actually find out what exactly it is. Either it is a homozygous dominant or a heterozygous dominant. In order to find that out, what we do is known as a test cross, in which we take the homozygous dominant, we take the dominant phenotype, plant or organism, and cross it with a recessive one, which will always have a genotype of small d or recessive homozygous recessive. So this helps to determine if the genotype this helps to determine the genotype of the dominant phenotype. For homozygous dominant genotype F1 progeny, that is if it was capital D, then all the progeny will have dominant phenotype. We'll get to the Punnett square in a bit, but if we have homozygous dominant, all the F1 progeny will display the dominant phenotype. But in case of a heterozygous dominant genotype, there will be equal amount of dominant phenotype and equal amount of recessive phenotype. So what exactly do I mean from this? So I would draw your attention to the left hand side of this graph, of this schematic. In case we have a homozygous dominant phenotype and we cross it with the recessive one, what we will get is that in the F1 gender progeny, we will get a heterozygous progeny, and the phenotype will still be that of the homozygous dominant. So cross between homozygous dominant and homozygous recessive will give you a heterozygous genotype, but the phenotype will still be that of the dominating parent. Thus, uh, the F1 ratio between the dominant phenotype and the recessive phenotype will be 1:0, or everything will be dominant phenotype. But in case of parent being heterozygous, this should have been heterozygous. We will get an F1 ratio of 1:1. That is, when you draw the Punnett square, you will see that the phenotypic ratio will become 1:1 for the phenotype. So we will have one organism which will show the dominant phenotype, one organism which will show the recessive phenotype. And thus, using this analysis and comparing the F1 generations, you will be able to say if the original organism was a dominant, heterozygous dominant or a homozygous dominant. So our current answer for this question is number B, that is test cross. Does anyone want to add anything to this? Okay. See, we go on to the next question, which is if Imtiaz is given another single blind plant to investigate whether the plant is a true breed or not, which of the following stands true for the condition? So in this case, we do not want to find out the exact genotype of the particular plant, but we only want to know if the plant is true breed or not. What technique can we use in order to determine that? The options are by crossing it with another known true breed variety. If any individual of different varieties were seen in the progeny, by extracting chromosome and checking under the microscope, by self-crossing, if any individual of different varieties were seen in the progeny, by test crossing if most of the progeny are of different types. So this is only one option correct type of answer. So Imtiaz wants to is given a plant whose genotype he does not know and wants to know if it is a true bred plant or not. That is, it has all homozygous traits or not. So which technique can he use? Only one option is correct. You can go ahead and try and answer this. Arushi says it is C, by self crossing. Anyone else that wants to try and answer this? Shea also says it is self crossing. Okay, so let us try and understand. So obviously everyone is correct this time also. It is a self cross is what is done to in order to determine if the genotype of the so it is self cross is used to determine if the genotype is homozygous, that is true bred or not. So what is self crossing? Self crossing is the cross between the same genotype or most of the time the same organism in case of plants that have both the that can be self-crossed within itself, such as the pea plant, and this is done in order to determine if it is a homozygous plant or a heterozygous plant. So what exactly is done is that you get you just self-cross with the same genotype. So if say the organism was homozygous, then one of its both of its alleles are homozygous dominant in both cases, and in the F1 generation you get again homozygous dominant itself. So identical phenotype in the F1 generation, which will tell us that the organism was homozygous to start with. So 100% of one trait. This is the Punnett square, which shows G, 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 and everything else is capital G, capital G. In case the organism is heterozygous, when we cross it, so it is a heterozygous trait, and when we cross it, we will see a mix 1:2:1 combination in the F1 generation, which will tell us that the organism was heterozygous to start with. So this exactly is the how we determine if the organism was true bred or not. So if Imtiaz is given a single blind plant to determine whether the plant is true bred or not, which of the following stands true? The option is C, by self-crossing if any individual of different varieties are seen in the project. So if by self-cross you see any different varieties, then the plant was not true bred. If you do not see 100% of, if you see only 100% of one phenotype, then the organism was true. So anyone wants to ask anything from this? I guess not. Okay. So we move forward to the next question. Which of the following is an example of incomplete dominance? The options are plant with parent progeny yellow and green pods producing yellow pods, plant with parent progeny yellow and green pods producing orange pods, plant with parent progeny yellow and green pods producing green pods, plants with parent progeny yellow and green pods producing half yellow and half green pods. So this is an one option type of correct answer. So everyone can try and put into one and put in chat one answer that they think is correct. Shreya and Arushi both say that it is B. That is, Ria also says it is B. That is, plant with parent progeny yellow and green pods producing orange pods. So anyone else wants to add anything to this? Any other answer? Nidhi says that the answer should be D, plant with parent progeny yellow and green producing half yellow and half green coats. Okay, so let us go and look into what exactly is incomplete dominance and try and answer this. So incomplete dominance is a form of gene interaction in which both alleles of a gene at a locus are partially expressed, often resulting in an intermediate or different phenotype. So what exactly happens is that in this case we do not have two forms of genes directly, that is dominant and recessive. In this case we might have that both the recessive trait, whenever it is present along with the dominant one, doesn't allow complete takeover in case of phenotype. So if for this example, when we in case of the most famous example is in the case of the snapdragon flower. In the snapdragon flower, the flower color red is the dominant phenotype and white is the recessive phenotype. And the red flower is denoted by capital R, and the white color is smaller, small. But when you cross, when we have a monohybrid cross in the F1 generation, we see a pink flower that is capital R, small r. That is, even though dominant trait is present, it does not express solely in the phenotype. When we take it to the F2 generation, we see the same kind of separation of traits that we see normally. That is a 1:2:1 combination. But in case of the heterozygous traits, the dominant trait does not take over, and we do not have a 3:1 phenotypic expression, but instead the phenotype also follows the genotype in 1:2:1, and so we have one red, two pink, and one white. So one red, two pink, one white flower color. So in case of incomplete dominance, we get a intermediate or blended form of the entire of the genotype. So the correct answer should be plant with parent progeny yellow and green pods producing orange pods. Option D is not correct, say producing half yellow and half green color, because when we segregate the different genotypes into individual, they then combine to form a separate genotype. So it does not happen that we see a chimeric kind of genotype, or even if say in this case if R and R was present, we do not see a chimeric phenotype, but instead we see one dominating over the other. That is what the laws of Mendel say. So the correct option is B. Any questions regarding this? Okay, so we move forward. A man with blood group B positive was married to a AB positive woman. Which of the following is true about the progeny's blood group? The child can be B positive, or AB positive, A positive, or O positive. The child can be B positive, AB positive, and A positive. The child can be B positive, AB positive, and O positive. And the child can be B positive, A positive, and O positive. Only one option is correct. So you can go ahead and try answering this. Instead, I will just read it. I am audible, right? Hello? Hello? Yeah, okay, thank you. So everyone is going for B positive, AB positive, and A positive. But since this is so, the blood group questions come a lot in various examinations, and these are important to understanding what is going on in the entire genetic scene. So this is the question that we will do by hand. So let me just go ahead and present my screen. Not do it. Screen should get shared. So let me just edit my. So in case of blood groups, there can be blood groups show another example of incomplete dominance, or rather codominance. In case of codominance, if we can have two dominant traits. So in case of blood groups, the two dominant traits are A and B blood groups. So the traits are given as IA and IB. These are the two dominant traits that can occur. So if when we say our person's blood group is A, what we mean is that the person's blood group is actually IA, or blood group is B, it can be IB. If the blood group is AB, then the genotype is IAIB. On the left hand side, these three are the phenotypes. On the right hand side, this is the genotype. Now, another thing can occur when the gene locus is getting divided in the chromosome. There can be a deletion or mutation that causes either A or B not to be present. That is known as the O blood type, or rather the O genotype, which is given as IO. But this is the recessive type, whereas both of these are dominant. So, in case of blood group A, it can be IA, and I have to admit some. Okay. So, in case of the A blood group, it can be IA, or it can be IO as well. Similarly, for for B blood group, it can be IO as well. And in this case, this is normal, the dominant trait A dominating over the recessive trait O and displaying the dominant phenotype blood group A. Is this clear till now? Yes, no, maybe, can be. This should be, this is clear, right? Okay. So whenever we are, so most questions that are asked from this particular type of blood group type of problems is that you have to choose the blood group of the progeny depending on the blood group of the parents. So we are given that the blood group of one of the parent is B. So that can mean either IB, or as we discussed, it can be IBIO. And the second parent has AB as the blood group. So it must be IAIB. So we essentially have two Punnett squares that we can draw from this. The first will have IB on one side, and another side we will have IAIB. So the options that we will from the Punnett scale itself, that gives us the phenotypes of AB blood group, AB blood group, AB blood group, and B blood group. Similarly, from this particular Punnett scale, again, if we draw, we will get we have IA, IB, we have a IA, IO in this case as well. IB, 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 I. So the phenotype in this case becomes AB blood group, A blood group, and a B blood group, which can be either homozygous B or heterozygous B. So the entire, we when we look into the question, which of the progeny's blood group can be possible, it can be AB, it can be A as well as B. So this should be the answer to our question. Going back to our slides we have. So going back to our slides, this is the question. So now we move forward, and this is a discussion on blood group genetics. That the blood group is determined by two alleles, IA and IB. Both alleles co-dominantly present together in the IA-IB genotype, and we have the blood group AB. If both alleles are absent, then we have the O phenotype, in which the genotype is IO-IO. So here we have a diagrammatic representation of exactly which genotype occurs with which. If both A's, we have A. If A and B, we have AB. If A and O, we have A. Similarly, AB, B and B, and A, B and O. So this is the particular problem. One of the parents had IBIB and IBIO as the genotype, and another had AB, which can only be IAIB. We performed the Punnett square analysis and found out that blood groups, in case the first parent is homozygous blood group B, then the blood groups can be A, B and B, and if he is heterozygous blood group B, then the blood groups can be A, AB and B. So finally, a man with a blood group B positive married to a AB positive woman can have child with B positive blood, AB positive blood group, and A positive blood. So this was the this is how exactly you have to solve these type of questions. So anyone has any questions regarding this? Anyone wants to add anything to this? I guess not. So what we will do is we will move forward to the next question. So we have another question on, okay, Archana says no, we are clear on this. Okay. So we have the next question on blood groups again. So the question is, if a couple with blood group B positive and O positive give birth to blood group B positive and O positive progeny with equal ratio. So parents, in the parental generation we have B positive and O positive, and in the progenies also we have equal ratio of B positive and A positive children. Then what are the positive genotypes of the parents? The options are IB-IB, IO-IO, IB-IO, IB-IO, and similarly other phenotypes as well. So which of the following is the correct one? Nidhi says it is C, IB-IO and IO-IO. Anyone else? Shreya also says it is C, IB-IO and IO-IO. Okay. So anyone else that wants to try and answer this? Okay. So we'll try again and share our screen and try to solve this one. Okay. So this is question number 7. This is question number 7, in which our parents have the blood groups B positive, which can again be either IB, both homozygous dominant, and IO, heterozygous. Or we have O positive, which is which can only be IO, nothing else. So this is our P generation. So we have two cases from here. We have two cases from here that can occur. The first one is that we cross IB into IO. Simple Punnett square analysis tells us that in all the four cases we have 1, 2, 3, 4 heterozygous B, which is denoted by IO. But in the second case, IO, we have uh, B and O in equal ratios, 2B and 2Aos. If you notice, so two heterozygous Bs and two homozygous O blood groups. So given the question, let us go back to the presentation. So if the couple has a child with equal, then what are the possible genotypes that the parents could have? So here we have, so blood group can be IB, IO, and for the O it could only be IO. And when we do the Punnett square analysis, we have one blood which will give 100% B blood group as the phenotype, and the other one which will have equal number of B and O phenotype, thus giving us that question, that the answer that the question asked, what a possibility for the B plus and O plus blood group progeny with equal ratio is the option C. If the one parent had heterozygous B blood group and the other has O blood group, IBIO and IOIO. So that was that question. Anyone has anything to add to this? Anyone wants to know anything about this? Okay, I guess not. So we move on to the next question. What is true for the blending hypothesis? That it is applicable on weight of the progeny, applicable on the gender of the progeny, applicable on the height of the progeny, and don't affect the progeny at all. Which of the following is correct in case of the blending hypothesis? You can go ahead and try and answer this and put the probable answer in the chat box. Which of the following is true for the blending hypothesis? It is applicable on the weight, gender, or the height. Archana says it is height. Or anyone else? Okay. So So inheritance hypothesis varied a lot before Mendel actually set in and put down his laws. So Shea also says C, which is height. So one of the famous blending hypotheses were that uh, the traits of the parents were the phenotypic traits of the parents blended and mixed together in order to form the traits of the next generation. That is, blending hypothesis says that parental contributions are combined. So before Mendel, traits were thought to be blended down and passed down. So if blending occurred, all the future offspring would be green. So if the blending hypothesis was correct, then we will always have one type of progeny or one type of trait that will get amplified throughout the generations. We will not have any trait that was present in previous generations reoccurring in the subsequent generations. So think about the dominant recessive versus a dominant. Uh, think about the homozygous dominant versus a homozygous recessive cross, and we know that in the F2 generation, the dominant recessive phenotype comes back, which will not be we will not be able to explain with the inheritance hypothesis, but with Mendel's laws. So this hypothesis was more or less moved aside when Mendel's laws were established. But in some cases, this hypothesis still works, like in case of the height of the individual, where there is a huge amount of genes that actually contribute towards the final expression or the final phenotype of the height. So essentially, in that case, it looks like as if the blending hypothesis is working because a lot of genes are working together, and those are not always follow the Mendelian laws because a lot of things are in there. So, if for the which of the following is true for blending hypothesis, it is applicable on the height of the gender. So, anyone wants to add anything to this? So, let me ask a question on this. If blending hypothesis looks like this, does this not look very similar to the incomplete dominance that we saw in case of Snapdragon? That we have the parent have one parent having a red phenotype, another having a white phenotype, and the progeny have a pink phenotype, which looks very much like the blending hypothesis is correct. So can anyone tell me why exactly that is, how exactly is incomplete dominance different from the blending hypothesis? So why do we have two different terms? Yeah, so Archana is spot on. So it differs in the F2 generation. So if we go back, so if I would like to draw your attention to the fact that if in the F2 generation, the blending hypothesis still suggests that the phenotype should be the same as the F1 generation, as there is inherent mixing of the parental traits already that has occurred. So the F2 generation should all be same. But if we go back, in this case, we see that in case of Snapdragon incomplete inherent dominance or any incomplete dominance, if the in the F2 we will have reemergence of the parental generations homozygous traits as well as the incomplete dominant heterozygous traits. So you have reappearance of the phenotypic trait of the P generation in the F2 generation. And in case of the blending hypothesis, this wouldn't have occurred and everything should have been pink. That is why incomplete dominance and the Mendel laws are the one that are now being followed and blending hypothesis has been casted aside. So that was correct, Archana. So that was question number 8. We move forward to the next question. This is the big question. So Imtiaz took another single blinded plant. Imtiaz took another single blinded plant. The plant is of self-pollinating nature. After self-pollinating for a few generations, he obtained pure breed of round and wrinkled pod-shaped plants separately. All the offsprings obtained by breeding pure breeds were all round pod-shaped plants. F1 generation was led to self-pollinate and produce 200 offsprings in the F1 F2 progeny. It was found that 150 were round and 50 were wrinkled. Help Imtiaz deciding the acceptance of Mendelian traits for the given plant. So what exactly did Imtiaz do? First, he got two separate plant types, one with round pod shape and one with wrinkle pod shape. These are pure breeds, that is, they should be homozygous in their traits. He crossed them to find the F1 progeny, which all of them had round pod shaped plants. And now he allowed this F2 progeny to self-pollinate amongst themselves and got 200 offspring, out of which 150 were round and 50 were wrinkled. So Imtiaz is wondering if this entire experiment follows the Mendelian pattern or the Mendelian laws. So the options that are given here are: the plants follow the Mendelian pattern as there is no blending of traits in the outcome. Plants do not follow Mendelian traits as there is no blending of traits in the parent's progeny. Plants follow the Mendelian trait as the progeny follow the proper segregation of the traits. Plants don't follow Mendelian traits as the progeny follow the proper segregation of traits. So only one option is correct. Shreya says it is plant C, that is plant follow the Mendelian trait as the progeny follow the proper segregation of traits. Anyone else wants to try? Archana says it is C as well. Plants follow the Mendelian traits as the progeny follow proper segregation of traits. Anyone else? Okay. So this is the easiest example of monohybrid cross that Imtiaz is trying to do. He got one homozygous trait. In this case, it is tall with homozygous capital D and dwarf with small t. In the first F1 generation, it will be a monohybrid capital T small t, which is essentially heterozygous for the tall trait. So every one of the plants should be tall. Like in his case also, every one of them were round. When he does selfing for the F2 generation, he will see that after combination, the phenotypic ratio will be 3 is to 1 and the genotypic ratio will be 1 is to 2 is to 1. Normal monohybrid cross. And since the phenotypic ratio is 3 is to 1, we can go ahead and see that it is indeed 3 is to 1 in the experiment he did. 150 round and 50 wrinkle. The ratio is 3 is to 1. So essentially monohybrid cross is what he did, and the plants did follow, the trees did follow the Mendelian pattern. But exactly what Mendelian pattern did they follow? Since they followed the law of segregation, because they were segregated one particular from one particular gamete, we get segregation into two different alleles that later recombine. So the correct option with Imtiaz with this entire experiment is plants follow the Mendelian trait as the progeny follow proper segregation of the traits. So anyone wants to add anything to this? Okay. So we go to the last question of today, which is a plant having a genotype A small capital A small a capital B small b or capital T C small c genotype was crossed with another plant with genotype capital A small b capital C. What is the probability X of an F1 offspring to be capital A, capital B, small b, and capital C, capital C? When the when this particular progeny offspring were self-crossed, what is the probability of another genotype, which is capital A, capital small b, capital C. So this is a probability type question, and we have various options. The first is x equal to 1 by 8, y equal to 1 by 4, x is 1 by 32, y is 1, y equal to 0, x equal to 1 by 8, y equal to 1 by 32, x equal to 1 by 16, and y equal to 0. Only one option is correct. So this is a good probabilities problem, and we will look into how exactly multi-hybrid process. In case of multi-hybrid process, you really do not need to draw a huge Punnett square. Just smaller 2 into 2 Punnett squares will do, and we can use just the laws of probability, which is essentially conditional probability, to solve this kind of problems. So Archana goes ahead and says that the option is D, x equal to 1 by 16 and y equal to 0. So I would encourage everyone else to try and answer this as well. Get a pen and paper and try this out. Just do the monohybrid process for individual traits A, B, C, and then find the conditional probability that this particular phenotype occurs or not. This particular genotype occurs or not. And same in the second case as well. You get a pen and paper, try to solve this. So the entire thing can also be extended back to dihybrid process. No need to worry about how exactly dihybrid drawing a Punnett square of 9 is to 3 is to 3 is to 1 in dihybrid process. You can still use this particular method of probability to solve those as well. Shreya also says that the answer is D, that is x equal to 1 by 16, y equal to 0. So please go ahead and try to solve this. So we'll wait another minute till everyone tries, and then we'll solve the problem. We have only 2 answers in chat. I expected more people to try and answer this. Okay, so let me share my screen, and so here we are back to the whiteboard. For this type of questions, the trick is that you have to simplify the entire multi-hybrid cross into simple monohybrid crosses. So the genotypes that we have are a small a, so this will separate as capital A small a, capital B small b, and capital C small c. Similarly, for the second one, we will have capital A, small b, capital C. This will separate as A, small b, capital C. So as I said, we separate them into smaller simplified crosses. So the first cross will be just A, small a, small a, capital A, small a. Similarly, the second cross will be B, small b, capital B, small b, capital B, small b, and the final will be capital C, small capital C, capital C, capital C, small c, capital C, small c. Okay. So this essentially gives us that the probability of capital A occurring is 1 by 4. Probability of capital A small a occurring is half, and probability of small a occurring is 1/4 again. Similarly, capital B occurring is essentially 0. Capital B occurring is half, and small b occurring is half. Similarly, C occurring probability is half, and small c occurring is also half. So here what we do is we calculate what is known as a conditional probability. That is, we have a condition that we have to fulfill, and we calculate probability accordingly. So conditional probability here is the probability of having A, B small b, C. So there is a rule known as which tells us that in case the probability is of independent events, then the probability can be divided into multiplication of those independent events. If we are finding the probability of events that are independent to each other, then we just multiply it to find the entire probability. This is the rule of probability that you probably would have come across. So the first one is 1/4 from here, the second one is half from here, and the third one is half again from here. So this gives us 1 by 16 as the first answer. Is this okay with everyone? Okay, Amardeep says it is okay. So next we have our question is, when the B, small b, capital C offspring were self-crossed, what is the probability Y? So this was the probability X, P of X. what is the probability y of a F2 offspring with genotype a small b small v capital C capital C? So we do nothing. We go back to a so this was our F1 project. This was our F1. So we are selfing this out. So again we go back and see what our traits were. They were capital A, small a, B, P, capital C, capital C. Oh no, sorry. Our F1 progeny is now capital A, B, small b, C and C, which will segregate as capital A, which does not matter much. We could have only written capital A, B, B, and again capital C, capital C. We are selfing it, selfing the entire thing. So this is just this cross, again another of the same type, which will give you again the same thing, capital A segregation, B small b, and capital C segregation. When we do our process, we again take individual independent traits because of the law of independent assortment. Everything should be capital A or homozygous dominant. Similarly for B and B, we have homozygous B, heterozygous B, heterozygous B, and homozygous recessive B. Similarly for C, everything is capital C. So homozygous dominant C. So our PY comes up to P, so probability, conditional probability of capital A, small v, small v, capital C, capital C. This will come up as we just discussed, law of independent probabilities. This will be multiplication of individual probabilities. Now comes the entire problem that till now it was all well and jolly. Problem occurs is that we need to find a probability of heterozygous A. But as our Punnett square declares, there is no possibility of heterozygous A. So this will be 0. For homozygous recessive B, we do have a probability of it, which is 1/4. And for homozygous dominant C, we have entire probability of 1. Everything should be homozygous dominant. But that does not matter. Multiplication to 0 gives us 0. So this is our F2 project. Thus the entire problem boils down to P of x is our 1/16, P of y is 0. So this was the entire problem. So any questions regarding this? How to solve multi-hybrid causes? So these are the most common types of questions. I would say most common are dihybrid questions and then followed by multi-hybrid ones. And these are simple to solve if you just know how to get simplify and then multiply back. Any questions regarding this? Okay. Okay. So this is what we just did. Wait, my screen is still not visible here. We'll go back and make our screen visible. The PPT is visible now, right? Yes. The PPT is visible, right? Presentation. Hello. Yes, I guess that it is. Okay, so this is what we just discussed. For the two genotypes for the parents given, the segregation will occur like this: individually segregated, capital A small a, capital B small b, capital C small c. Second one will have capital A small b, capital C capital C. This is our P generation. We do the Punnett square for only the A trait. We get the segregation such as this: one fourth A capital A, half capital A small a, one fourth small a small a. Similarly for B, and finally for C. And then we calculate the probability of P capital A, capital B small b, capital C capital C. Capital A only occurs 1 out of 4 times, one fourth. Capital B small b occurs half the time, so 2 out of 4, half. And capital C occurs again half the time, 2 out of 4. Multiply all of them, we have 1 by 16 as our F1 progeny. A probability of F1 progeny having this particular genotype is 1 by 16. We follow that up with selfing this F1 progeny. The segregation will again occur similarly: capital A, capital B small b, capital C for both of the cases. We draw the Punnett square for A, which gives homozygous A throughout; B, which gives 1 is to 2 is to 1 segregation for B trait; and for C, it is homozygous C again. We then go ahead and find out capital A small a probability, which is 0 in this case. Then it does not matter we find anything else because multiplication with 0 is 0, but still for small b it should be one fourth, and for this it should be 1, and the answer is 0. It should be one fourth, and this should be 1. And doesn't matter what we multiply with, because the final answer always comes to 0. Multiplication by 0 always comes to 0. So the answer to our last question is: what is the F1 probability of X having F1 offspring with genotype capital B small b, capital C, that is 1 by 16, and after self cross, which is the probability of capital A small b, capital C, that is 0. So that was this question happens to a probability to solve multihybrid process. Any questions regarding this? Anyone wants to ask anything? Okay. So if not, that was today's session. Since we have time left, we can discuss anything regarding week 4 or before in this particular session. Anything you want to ask, you can just go ahead and put it to the chat box. You can discuss. We might even go through the PowerPoint slides of the course if we have any doubt regarding. Doesn't Yes, Archana, you can go ahead and type your question or anything you want to ask into the chat box. No, sir. Okay. So if you do not have any questions, then you can just leave the meeting, and then we will meet next session in which we will. What are the Amadou says thank you. Archana asks what are the priorities for PMRF. So there is a website, there is an entire national website out there, PMRF.in. You can just go ahead and check the entire priorities, prerequisites, what are the various things that we have to do about reviews, contingency grants, everything that is on, or everything is all on the site. PMRF.in. Anything else regarding the course itself? Okay. Okay. So if you have any questions, you can stay and ask. If you do not, you can just leave the session. Okay, I think no one had anything to ask because everyone has left. So that will be end of today's session. We discussed various basics of genetics and solved various problems on mathematical problems on genetics, as well as discussed various basics on laws of Mendel's, as well as monohybrid, dihybrid, and multihybrid process. So that is it for week 4 PMRF-NPTEL lecture on the course bioengineering. On the course bioengineering. So I thank you all for joining and see you all next week. Thank you.